The consequences of point mutations. So what are point mutations? Point mutations are a mutation that alters a single base position in a DNA molecule by converting it to a different base or by inserting or deletion of a single base in the DNA molecule. There are three types of these mutations. The first is situational, then there's insertion, and finally deletion. To the side, you can see an example of each, and continuing on, we will discuss specifics. The first mutation we will talk about is base substitution. This is a mutation in which one base pair is replaced by another. There is also two subtypes. The first being transition. These subtypes are defined as a replacement of a base by another base of the same chemical category. An example of this is a single base DNA strand. In a single base DNA strand, a purine is replaced by another purine, or pyrimidine is replaced by another pyrimidine. In a double-stranded DNA, both members of a base pair in the same relative location must have the same change. The second subtype is transversion. This, which are a replacement of a base of one chemical category by a base of another. An example of this would be a purine replaced by a pyrimidine, or a pyrimidine replaced by a purine. To give a real-life example, we can look at the cause for sickle cell disease. In this disease, a glutamic acid is substituted with a valine, causing this conformation change in the cell. Next, we could talk about the mutation of substitution. All of these mutations are direct consequences of two aspects of gene codons, or codes. The first is degeneracy of the code, and the other is the existence of translation termination codons. There are three types of these mutations. The first type is synonymous mutations, or silent mutations, and they never alter the amino acid sequence of a polypeptide. This mutation changes one code for an amino acid into another codon. This mutation changes one codon for amino acid into another codon for the same amino acid. The second type of mutation is called missense mutations. This mutation is defined by a codon for one amino acid being changed into a codon for another amino acid. Amino acids can be changed by conservative substitution or non-conservative substitution. In a conservative substitution, it may replace one amino acid with a chemically similar amino acid, and this alteration is less likely to affect the protein structure and function severely. In non-conservative substitution, one amino acid may be replaced by a chemically different amino acid, and the alteration is more likely to produce severe changes in protein structure and function. The final type of mutation is nonsense mutations. These mutations are defined as a codon for one amino acid being changed into a translation terminating codon or a stop codon that can lead to premature termination of the translation and have considerable effects on protein functions. One of these products may lead to a complete inactivation of the protein, which may be due to a splice site mutation. This can dramatically change the mRNA transcript by leading to larger insertions or deletions that may or may not be framed. Example of these mutations you can see on the slide under transcription or transversion. Base pair insertions or deletions are also called indel mutations. These mutations may have consequences on polypeptide sequences that extend far beyond the site of mutation itself. An example of this is the addition or deletion of a single base pair of DNA that changes the reading frame for the remainder of the translation process. This would make the site of a base pair mutation to the next stop codon in the new reading frame and make the mutation called a frame shift. Frame shifts cause an entire amino acid sequence translation downstream of the mutation site to bear no relation to the original amino acid sequence and cause loss of normal protein structure and function. These types of mutations also bear a resemblance to the CRISPR-Cas system. CRISPR targets a specific sequence of DNAs or RNAs that can delete and insert or even substitute new genes or an amino acid into the cell. In research, CRISPR can be used in areas such as cancer research. I was able to find a research article called Point Mutation in Essential Genes with Loss or Mutation of a Second Allele by Beck and Jester and colleagues. Scientists studied the use of point mutations to understand the importance of specific ribosomal protein genes in cells for anti-tumor immunity. This study was done in the early 2000s before CRISPR research became a focus, so it was not utilized in this research. But if we did this experiment again, it could provide more insights. Along with consequences of coding regions, there's also consequences in non-coding regions. Within non-coding regions, they do not directly encode a protein, but do contain many crucial DNA binding sites for proteins interspread among sequences that are not essential for gene expression or gene activity. On the DNA level, binding sites include the sites in which RNA polymerase and its association factors bind, as well as sites to which specific transcription regulating proteins bind. At the RNA level, additional important binding sites include the ribosome binding site of bacterial mRNA, the 5'3' splice sites for exon joining the eukaryotic mRNAs, and sites that are regulate translation and localize the mRNAs to particular areas and compartments within the cell. There's a few causes to these point mutations, the first being errors in DNA replication. As you can see in the picture below, the non-coding strand of DNA will go on to continue replication as normal. But the coding strand of DNA, there will be changes in base pairs, causing a point mutation to happen, as you can see in blue. The second cause for mutation is errors in DNA recombination. This is caused by DNAs becoming lost during replication. The next cause for mutation is chemical damage to DNA. There's many chemical changes that can happen to cause mutations. Some can be exogenous, others man-made, and there are still some created by the environment. The last cause for mutation is radiation. These mutations generate free radicals that can cause chemical damage to DNAs. To regulate damage control of DNAs, proteins like P53 and P21 are activated. Once a signal is sent out by the cell, protein P53 is phosphorylated and activated to bind to the regulatory region. of the P21 gene. This activates P21, which is a CDK inhibitor protein, and causes a conformational change in the CDK complex. This inactivates it and results in cell cycle arrest. If P53 function was lost, it can cause severe cell damage and possible cell death for the cell. In about 50% of all human cancers, they have the mutation of P53 gene, which inactivates it. Example of diseases. Finally, we can discuss some examples of diseases caused by point mutations. The first is cystic fibrosis. This is an inherited disease characterized by buildup in thick, sticky mucus that can cause damage to many of the body's organs. This is caused by many mutations, but the most common one is the deletion of 3 nucleotides in the cystic fibrosis transmembrane conductance regulator gene that results in the loss of an amino acid and causes an incorrect folding in protein. The second disease caused by point mutation is sickle cell anemia. This is also an inherited disease in which there is not enough healthy red blood cells to carry oxygen throughout your body. This mutation is caused by a substitution of a valine to a glutamic acid. Lastly, here are the references I use to do this study. Thank you.